Hello guys, today we are going to learn enzyme kinetics, which is important topic in bioprocess engineering for GATE biotechnology students. So first we will see what are enzymes. So enzymes are actually protein molecules that help to speed up metabolism or the chemical reaction in our bodies. Okay, so they are actually a sort of biocatalysts. Biocatalyst means these are those catalysts present within the biological systems. Okay, so that help to speed up metabolism, that is they increase the rate of metabolism or the chemical reaction that occur within body. So enzymes are sort of biocatalysts which are required for all the metabolic reactions present in our body. Alright, so such is the importance of enzymes. Mostly they are protein molecules, but there are few exceptions, like we have ribozyme. So ribozyme is not a protein molecule, although it is an enzyme. It is actually an RNA molecule. So you have to remember this example. Questions are sometimes asked that name an enzyme which is not a protein molecule, then you have to write ribozyme. Ribozyme is also an enzyme, but it is not a protein molecule. It is actually a catalytic RNA molecule. Okay, so except ribozyme, almost all proteins that help to speed up metabolic reactions within the biological systems are called as enzymes. Now let's see what happens in an enzymatic reaction. So in an enzymatic reaction, apart from enzymes, you need two things. One is the substrate and the other is the product. So first, one theory suggests that enzyme combines with the substrate to form an intermediate called as the enzyme substrate complex, and this enzyme substrate complex later on breaks down into enzyme, free enzyme and the product. Okay, so there are two reactions occurring simultaneously whenever an enzyme is performing a biological reaction. So first, enzyme combines with the substrate to form enzyme substrate complex. So the formation of enzyme substrate complex is comparatively faster. Okay, so the formation of enzyme substrate complex is comparatively faster, and then the enzyme substrate complex dissociates into free enzyme and the product. This reaction rate is comparatively slower than the first. Okay, so these are the chemical reactions or biological reactions that occur whenever an enzyme is performing a metabolic reaction within the living system. So here, as you can see, K1 denotes the rate constant of the forward reaction, that is the rate constant of the formation of enzyme substrate complex, and K minus 1. So minus 1 suffix denotes the rate constant of the backward reaction, that is dissociation of enzyme substrate complex into enzyme and substrate. Now let us see the function of enzymes. So the function of enzyme is basically to lower the activation energy. So activation energy is what? Activation energy is the minimum amount of energy required by a reactant molecule to get converted to the product. So it is the minimum energy, it is a barrier, minimum energy barrier. When the reactant crosses that minimum energy barrier, then it can be converted into the product. This is called as activation energy. So enzymes, what do they do? Basically, the function of enzyme is lowering the activation energy. So the main function of enzyme is lowering the activation energy. Okay, so the main function of enzyme is lowering the activation energy. So here you can see no enzyme. When no enzyme is present, the activation energy is so high from here to here. Okay, and in presence, let us see what happens in presence of the enzyme. When enzyme is present, the activation energy has been reduced. Okay, so here you can see, so activation energy is reduced by this amount. Activation energy has been reduced. So as you can see, in absence of enzyme, activation energy was much higher in an uncatalyzed reaction, but in presence of enzyme, activation energy has been reduced to a very small extent. So the function of enzyme is lowering the activation energy so that more number of reactant molecules can be converted to the product. So if you lower the activation energy with the help of enzyme, what will happen? More number of enzyme molecules will rush and cross the activation energy barrier to get converted into the product. So this is the main function of enzyme. Now let us see the derivation of enzyme catalyzed reaction, the equation which we call Michaelis-Menten equation. So first we have two reactions occurring in an enzyme catalyzed reaction. So the formation of enzyme substrate complex ES complex from free enzyme and substrate. So E plus S gives you ES complex, which is the enzyme substrate complex. K1 here denotes the rate of formation of enzyme substrate complex of the forward reaction, and K minus 1 denotes the dissociation of the enzyme substrate complex into free enzyme and substrate. It is the rate constant for the backward reaction. Okay, so this is one reaction. Another reaction, after formation of enzyme substrate complex, what happens? The enzyme substrate complex gets dissociated into free enzyme and the product molecule. So here K2 denotes the rate constant of the forward reaction and K minus 2 denotes the rate constant of the backward reaction. Alright. So if we combine that, then we have E plus S, K1 and K minus 1, ES, which is the enzyme substrate complex, then K2, E plus P. Now I have already told you the formation of enzyme substrate complex is comparatively faster, and the formation of the product from enzyme substrate complex, the second reaction, is comparatively slower. Also, generally, the formation of enzyme substrate complex from free enzyme and substrate molecule is a reversible reaction, and the formation of product molecule from enzyme substrate complex is generally an irreversible reaction. Okay, so that's why here we have two rate constants, but here we have only one rate constant K2, because the second reaction is generally irreversible. So this is our first equation. Okay, this is our first equation that we are having. Then we can also say that if we find out the velocity or the rate of dissociation of the enzyme substrate complex, then we will write it as K2 into ES, where K2 means the rate of dissociation of the enzyme substrate complex to form the product, and ES means the enzyme substrate complex within third bracket, which means it is the denoting the concentration. Okay, so ES within third bracket means the concentration of the enzyme substrate complex. So if you remember in class 12 chemical kinetics, we write rate is equal to rate of any chemical reaction. We write it as K, where K is the rate constant, and then we write the concentration of the reactant molecule, okay, within third bracket, denoting concentration. So here also we have written the same thing. Rate is actually a sort of velocity of the reaction. So V0 is K2, where K2 is the rate constant of the dissociation of the enzyme substrate complex into product, and ES is the reactant here, and written within third bracket means it is denoting the concentration of the enzyme substrate complex. So this is our second equation. Okay, this is our second equation. So rate of ES formation. So the rate of ES formation means now we are going to find out the rate of formation of enzyme substrate complex. Okay, so what will happen? So it is written as K1 into ET minus ES whole into multiplied by S. So ET means ET is the total enzyme concentration, and ES means the enzyme that is already bound to the substrate molecule. So ET minus ES will give you the free enzyme present that is now capable of binding to the substrate molecule. Okay, so ET minus ES is actually what? The free enzyme that is still present within the reaction system, which can still bind the substrate molecule. Okay, so ET means the total enzyme concentration, ES means the enzyme that has already bounded, with the substrate molecule. So ET minus ES denotes the free enzyme that is capable of binding to the substrate molecule. Okay, and whole multiplied by S. Alright, so if you multiply the rate constant with this whole expression, then you actually get, which is a rate of ES formation. Okay, rate of ES formation. So rate of ES formation, enzyme substrate complex formation, is K1, which is the rate constant, into the free enzyme present within the reaction system multiplied by S, concentration of the substrate. That will give you the free enzyme that is capable of binding to the substrate. Okay, so this we have another equation, which is equation number 3. Now we are going to see the rate of breakdown of the enzyme substrate complex. So we write it as K minus 1 into ES, that is the rate constant of the backward reaction multiplied by the enzyme substrate complex concentration, plus K2 into ES. Okay, why? You see, in this equation we are going to see ES breakdown, rate of ES breakdown. So in this equation, as you can see, K minus 1, if you take K minus 1 into consideration, it is actually the rate constant of the backward reaction, where ES is to be broken into free enzyme and free substrate molecule. Okay, so K minus 1 is actually involved in the breakdown of enzyme substrate complex in the first reaction. In the second reaction, it is K2. This is the reaction rate constant which is involved in the breakdown of the ES into free enzyme plus product molecule. So we have two rate constants. One, we have K minus 1, which is involved in the formation, which is involved in the breakdown of the ES into free enzyme plus substrate, and another rate constant, which is K2. This is also involved in the breakdown of ES into free enzyme plus product. So we have to take the two rate constants into account while we are going to determine the rate of ES breakdown. So one, we write it as K minus 1 into ES plus K2 into ES. Okay, this is our fourth equation. Alright, this is our fourth equation. Now we are assuming something which is called as the steady state assumption. So what we are assuming in steady state assumption is that the formation of enzyme substrate complex is equal to the breakdown of the enzyme substrate complex. So in steady state assumption, what we are assuming, the rate of formation of enzyme substrate complex is equal to the rate of breakdown of the enzyme substrate complex. Then what will happen? These two equations will become equal to each other. So we have done that. K1 into ET into E minus ES whole into S is equal to K minus 1 into ES plus K2 into ES. Now what we are doing, we are just multiplying, now what we are doing on the right hand side, we are taking ES common from the first step, and on the left hand side we are keeping every term as it was, and we are taking this, and then the next step we are taking this ES to this side on the right hand side. Okay, so because we want to take every ES common, so what will happen, K1 into ET into S, okay, is equal to K1 into S plus K minus 1 plus K2 whole multiplied by ES. This is our, say, fifth equation. Now from the fifth equation, we can write it as ES is equal to K1 into ET into S by whole divided by K1 into S plus K minus 1 plus K2. So this is our sixth equation. Now what we are doing, we are dividing both the numerator and the denominator by K1. Okay, so we are dividing both the numerator and the denominator by K1. So what will happen? ES will become ET into S by S plus K minus 1 plus K2 whole divided by K1. So we are dividing both the numerator and the denominator in equation number 6 with K1. So we get equation number 7. Now this term K minus 1 plus K2 by K1, this term is called as, or it is written as Km, where Km is called as Michaelis-Menten constant. Okay, so Km is actually K minus 1 plus K2 by K1. Now this relationship is very important for calculation of problems on bioprocess engineering. Okay, so suppose in the competitive exams in GATE biotechnology, many a times you won't be getting any other value. Many a times they just give you K minus 1, K2 and K1, and you need to calculate Km. Then you have to remember this formula. Okay, so Km is actually what? K minus 1 plus K2 by K1. So in the next equation, what we are doing, we are replacing this term K minus 1 plus K2 by K1 by the new constant called as Km, where Km is a Michaelis-Menten constant. So this is written as, this is our equation number 8. Now if you remember, we wrote it previously, V naught is equal to K2 into ES. Okay, we wrote it previously. If you have forgotten, please see the video once again. We have written that V naught is equal to K2 into ES. We have written it earlier, where V naught is the rate of formation of the product. Okay, K2 is the forward rate constant, where ES was broken down into product, and ES is the enzyme substrate complex concentration. We have written it that. Now ES is, we have got ET into S by Km plus S. So we substitute it here. So we will write it as V naught is equal to K2 into, what will we write here? K2 into ET into S by Km plus S. ET into S by Km plus S. So we will substitute it here, the values of ES that we got from equation number 8. Okay, so V naught comes to be K2 into ET by K2 into ET into S by Km plus S, where ET is a total enzyme concentration. Now K2 into ET, this term is often denoted by the term called as Vmax or Vm, where Vm is the maximum velocity of the enzyme catalyzed reaction. So this was our equation number 9. Alright, so the final equation is this one. V naught is equal to Vmax into S by Km plus S. Okay, so this equation is called as Michaelis-Menten equation. Okay, so this is the famous equation. Alright, so this was our equation number 10, and Vmax or Vm that we got from the previous step is actually K2 into ET, total enzyme concentration. ET is the total enzyme concentration. Now this formula is also very important like Km. Sometimes they don't give you to directly calculate Vm. Instead, what happens, they give you K2 and they give you total enzyme concentration. Then you have to calculate Vm with this formula only. Alright, suppose you are given, okay, suppose this is the Michaelis-Menten equation. V naught is equal to Vmax into S by Km plus S. So if you are given Vmax, Km and S, you can easily calculate V naught. But suppose you don't have any direct values of Vmax and Km. Instead, what they have done, they have given the values of K minus 1, K1 and K2, and they have given total enzyme concentration. Then, and also the total substrate concentration. They have asked you to calculate V naught. So what you will do, first from this formula, Km is equal to K minus 1 plus K2 by K1. From here you will calculate Km. Then from this formula, Vmax is equal to K2 into ET. You will calculate Vmax, and then you will put everything, the calculated value, in this formula. Okay, so you will put the value of Vmax calculated from here. You will put the value of Km that you have calculated from here, and you will put the substrate concentration. Then you will get V0. So sometimes you don't get any direct value. Instead, they give you indirectly the values of K-1, K2, and K1. So you will have to go in that way. So in that process, this formula, these two formula becomes very important for gate biotechnology. Now let us see what will happen for some case. Suppose we take V0. Okay, so suppose what will happen, let us see. Suppose we take V0 as Vmax by 2. Suppose we take V0 is equal to Vmax by 2. Then what will happen? In place of V0 we have written Vmax by 2. So is equal to Vmax into S by Km plus S. So Vmax, these two Vmax will get cancelled out. And then we have half is equal to S by Km plus S. So by transposition we will get Km is equal to S when V0 is equal to Vmax. So this is the case. That means what? When the velocity will attain half the maximum velocity. Okay, so when the velocity of the enzyme catalyzed reaction will be half the maximum velocity, the Km will be equal to the substrate concentration. So the Michaelis-Menten equation will be equal to the substrate concentration. Here, as you can see, this is a hyperbolic curve. This is a hyperbolic curve, and it is actually the representation of my graphical representation of Michaelis-Menten equation. So if you plot the Michaelis-Menten equation, the curve that you should get should be this hyperbolic curve. Okay, so this hyperbolic curve is plotted by plotting the velocity on the y-axis and the substrate concentration S on the x-axis. So if you plot in this way, you will get a hyperbolic curve. Now, as you can see, as V increases and attains Vmax, at this position, you get a substrate concentration, and which is actually the value of Km, or the Michaelis-Menten constant. Because why? From the Michaelis-Menten equation, if you put the value of V0 as Vmax by 2, you get Km is equal to S. That means the substrate concentration becomes equal to the Michaelis-Menten constant when the velocity will attain half the maximum velocity. So here, when the velocity is attaining half the velocity, the corresponding value that you are getting on the x-axis is actually the value of the Michaelis-Menten constant. Alright, so from the graph also you can plot and you can find out the maximum velocity. From there you find out the half of the maximum velocity, and the corresponding value on the x-axis will fetch you the value of the Michaelis-Menten constant. So from the graph also you can find out the Michaelis-Menten constant. Now as you can see, this Michaelis-Menten curve has actually two portions. One is this portion and the other is that portion. This portion is actually a representation of the first order reaction rate. Alright, this position is actually a representation of the first order reaction rate where the velocity increases with substrate concentration, and this portion is actually a representation of zero order rate equation. Okay, zero order rate equation. Why? Because here the velocity of the enzyme catalyzed reaction does not depend on the substrate concentration, because the system has already become saturated with the substrate. That is why if you increase the substrate concentration, because the enzyme molecules are limited, so the reaction rate will not increase. Even if you increase the substrate concentration, the reaction rate will not increase. So here what is happening, the reaction rate or the velocity is not dependent on the substrate concentration. So here it is representing zero order rate equation, and on the lower side this is dependent on the substrate concentration. So it is actually a first order rate equation. It is representing a first order rate equation. Why? As you can see, I will explain it here. Suppose you have very low enzyme concentration. Okay, so very low enzyme concentration. So what will happen? Enzyme concentration is very low, alright, and substrate concentration is very high. So what will happen? Most of the substrate will be in the free state, okay, and there will be very little formation of enzyme substrate complex because you have enzyme concentration very low and substrate concentration is very high. Okay, so this is a case where there will be almost no or very little formation of enzyme substrate complex. Suppose you have another case. Okay, so suppose you have another case where the enzyme concentration is very high and the substrate concentration is very low. Okay, so substrate concentration is very low and the enzyme concentration is very high. So what will happen in that case? Because the substrate concentration is very low, almost all the enzymes will be in the free state because you don't have that much of substrate to combine with the enzyme, and almost all the enzymes will be in the free state, not bound to any substrate, and there will be very little formation of enzyme substrate complex also. Now if you start increasing the substrate concentration, what will happen? There will be formation of enzyme substrate complex, and till you use all the available enzyme molecules. Now if you increase the substrate concentration beyond that way, that is the substrate concentration becomes very high, because the enzyme molecules are limited, what will happen? The reaction rate will not increase because the enzyme concentration is very limited. You have not increased the enzyme concentration proportionately with the increase in substrate concentration. So because the enzymes can combine with a limited amount of substrate, even if you increase the substrate concentration beyond the threshold, so the rate equation or the rate of the enzyme catalyzed reaction will not increase. Okay, so that is the main reason in first order reaction rate, in the initial stages of the Michaelis Menten graph, what is happening is that the enzyme concentration was very high initially and the substrate concentration was very low. Now when you are increasing the substrate concentration, what is happening? More and more enzymes are able to combine with the substrate and the reaction rate is very fast. That is why here you have first order rate equation where the velocity of the reaction rate is increasing with very little change in substrate concentration. Okay, up to here more or less you have first order reaction rate representation. Afterwards, when the enzyme molecules are becoming saturated with the substrate beyond the threshold, when you are continuously increasing the substrate concentration, what is happening? The reaction rate is not increasing, and that is why here it is approaching an asymptote. Okay, so here it is approaching an asymptote because the enzyme molecules are limited and you are increasing the substrate concentration. So what is happening? The enzymes are becoming saturated with the substrate and the reaction rate is not increasing even if you increase the substrate concentration. So from here to here, this is called as zero order reaction rate representation, because the rate of the reaction is not dependent now on the substrate concentration. So you have two stages in a Michaelis Menten graphical representation. In the initial stage you have first order reaction rate representation. In the second stage you have zero order reaction rate representation.